Awesome. Well, thank you everyone for joining our discussion today on single cell. We're super excited to have this discussion with you guys today. So today's conversation, of course, will be focusing on single cell as it relates to everything in the landscape. My name is Hannah Schuyler. I'm a director of business development here at g.nome. We also have our senior director of product management, Kit Furman, joining us, as well as Nora Kearns, our in-house single cell expert in bioinformatician. So super excited to have our panelists today for the discussion. So just a few quick things before we get started. This fireside chat is being recorded, and we will send it out to all attendees and registrants after the session wraps today. And then please enter any questions that you have for myself or Nora or Kit in the chat box, and we will address those at the end during a live Q&A session. So please feel free to enter them in, and we will discuss those at the end during our live Q&A session. So what will we be discussing today? We will be talking through everything as it relates to single cell. So what's new and exciting in terms of why single cell is important and what that means for researchers. We will also talk through some of the challenges that researchers face when analyzing single cell data. I know, of course, analyzing large data sets is sometimes complex for researchers. So we'll be talking through some of those complexities. And then we'll also be talking through some of the advancements. So, of course, single cell is important, but then researchers want to take it one step further in doing multi-omics or spatial analysis. So we'll be touching on that as well. And then lastly, we will have some few minutes at the end for live Q&A. So definitely want this to be interactive. If you have questions, please feel free to put them in the chat box and we will address those at the end. So just briefly, before we get into the discussion, I wanted to give you guys an overview of what Almaden Genomics is and who we are. So we are a software company that spun out of IBM Research Center in Almaden, California, hence the name Almaden Genomics. So the software was originally built as an internal need for pet and food safety. And the team quickly realized that there was a need and value for building pipelines and workflows for customers that may not know code. And so that's how g.nome was born. So over the last year, we've been working with a variety of different biotech, pharmaceutical, and academic clients to help researchers process and analyze their data. And what are we trying to do here at Almaden Genomics? I think one of the biggest things that we are focused on doing is really bridging the gap between both users, being the biologist and the data scientist and the bioinformatician. I'm sure many of you know that a biologist's goal typically is to analyze their data in experiments. They have either in-house sequencing data or publicly available data, and they're really focused on interpreting that data, making visualizations, and making sense of the data. So being able to do that without knowing code or how to build pipelines is super important for them. And they often are relying with bioinformaticians to do the analysis for them. And this can be a very time-consuming process. And there's not always a common language between the biologist and the bioinformatician and the data scientist. And then on the other side of that coin is the bioinformatician and the data scientist, right? They are really focused on building pipelines, workflows, leveraging open source tools. And their biggest thing is helping the biologist to analyze their data. So with that, we are really focused on trying to bridge the gap between both the biologist and the data scientist and bioinformatician. So on g.nome, we have a variety of different pre-built workflows that a user can access. And so today's discussion, of course, is focused on single cell. And there's many ways that you can actually process single cell data on the software. One of that being publicly available data through our SRA integration. So you have the ability to pull in public data, process that through our SRA tool from start to finish. You can also process in-house sequencing data that you're working on. So whether that's pulling data from a local file, a local server, or an S3 bucket. So you can process large amounts of data in that way as well. And then the third component of that is some of the data partners that we're working with. So we're working with a variety of data partners that provide harmonized and curated data sets that can be supplemental to the data that you guys are working with. So all of that can be processed through our single cell workflows. And then the goal is that you can actually utilize one of our pre-built workflows. So we're using Seurat and Star Solo for our workflows. So you can start with that, upload your FASTQ file, bring in your metadata file, and go start to finish with those visualizations. Or you can make modifications to the workflow as well. So there is that custom aspect of it where you can do batch correction, you can adjust normalization, filter out cells. We really want to provide that custom layer to our researchers as they're looking to analyze their single cell data. And then you can also create custom visualizations as well. So we do have Jupyter notebooks integrated into the platform. So this is, again, allowing you to create those custom visualizations. And then the last component of that, of course, is creating and sharing reports. So we do have interactive plots and reports that can be shared with your colleagues as they are looking to visualize and analyze the single cell data. So that's kind of a quick overview of Almaden Genomics and our software g.nome. And so I would like to kick off this fireside chat and discussion with a poll for the audience. So you should see it pop up in just a second. So if you could go ahead and answer this, are you working with single cell data right now or planning to in the near future? Either yes, I'm working with single cell data now, not yet, but I'm planning to in the near future, or no, I'm just curious to learn more. Give it a couple minutes for the responses to come in. Awesome. So it looks like we actually have a mix across the board with people working with single cell data, as well as some that are not yet and are just curious to learn more. So I think this is a perfect segue to kick off our discussion with Nora and Kit. Nora, I know that you are our in-house single cell expert. Do you want to give us an overview of single cell sequencing and why this is such a revolutionary technique in the space?
Yeah, of course. So single cell RNA sequencing refers to the process of sequencing the transcribed RNA from individual cells. And this is really useful because let's say you have a sample of lung tissue that you're interested in exploring, and you want to identify the genes that are expressed in that lung tissue. Well, there's multiple different cell types in lung tissue, and in traditional bulk RNA sequencing, we would only be able to find the average gene expression across the entire population of cells in that lung tissue. But with single cell, cells are basically isolated in little individual droplets before we sequence them. So we are able to measure gene expression in each individual cell in the tissue sample. So within the lung, you would be able to see how epithelial cells, immune cells, and nerve cells all express varying levels, even within their own cell type. So how does each individual epithelial cell express differently? So the analogy that's frequently used to describe single cell compared to bulk RNA-seq is that of a smoothie versus individual fruit. So when you're doing bulk RNA-seq, you're basically creating a smoothie, and you might not be able to tell where each kind of flavor is coming from. But when you have the individual pieces of fruit that went into that smoothie, you know, okay, so the sweetness came from the strawberry and the acidity came from the lemon or something like that, I think is a good analogy for it. And single cell is, again, very useful because profiling the gene expression activity in cells is really considered one of the best approaches to understanding cell identity and cell state, as well as function and then response to different stimuli, which might be really relevant in terms of drug discovery and disease understanding.
Yeah, that's great. And you did touch on this briefly, but what are researchers really trying to uncover from a scientific standpoint when they're looking at their single cell data?
Yeah, so I kind of think of this as having two sides, which is the translational side, as well as the research and discovery side. So I think translation is what's really most relevant to the general population and to medicine. And I think single cell has the ability to really advance personalized medicine. So for example, in cancer research, single cell can reveal the existence of subpopulations of cells within perhaps a solid tumor that might have different mutations or different response to treatment. And that can help doctors really understand, why individuals respond differently to therapies, and help them develop personalized medicine strategies. And I think a good example of this is we recently read a paper at Almaden on breast cancer research and using single cell to understand breast cancer tumors. And traditionally with breast cancer diagnostics, there's only a few diagnoses of breast cancer, how we classify tumors. But when you use single cell to explore individual tumors from patients, even within the same kind of diagnosis category, so all these tumors are all triple negative breast cancer, we see that there's actually massive diversity between those tumors. And that can really help us understand why one patient might not be responding as well to a certain type of therapy as another. And it helps us move towards better personalized medicine. And then I think from a research and discovery standpoint, for people like myself, single cell can help us enhance our understanding of biology overall and create resources that will help the field accelerate research and discovery. There's one initiative I'll speak to in particular, which is the Human Cell Atlas Project. The Human Cell Atlas Project is an international collaborative consortium with the objective of basically charting all of the cell types in a healthy human body from childhood to adulthood. So that would just be a fantastic resource for the field to have kind of a high resolution catalog of cells. For a number of reasons, it would facilitate our understanding of disease mechanisms. It would be an educational tool. And also as an open source tool, it would really help democratize research. So labs don't have to spend a lot of money doing more basic experiments just to get kind of an idea of what cells they're looking at. It would just kind of, you'd have that resource available. So I think it's really valuable from those two perspectives of translational medicine, and then also as, in terms of basic and discovery and research.
Yeah, I definitely would agree with that. And I'm sure, Kit, a lot of this resonates with what you're hearing from your customers. Can you maybe expand a little bit upon what you're hearing from our customers when they're reaching out about single cell?
Yeah, absolutely. People are doing such complex experiments now, whether they're in the preclinical space and doing, research, or they're close to the clinic and studying, endpoint data from clinical trials. There's so much analysis that needs to be done, and it's such a critical component of the scientific workflow. But we're all struggling to find time, the right collaborators, or the right software tools to analyze this data. And as Nora stated, single cell has become an important component of all of these different types of studies. And so analyzing the single cell data is a real challenge for the whole community. And that's why people come to g.nome to find solutions to those problems, right? They're trying to right now splice together different solutions from different software packages or open source tools. What I hear from customers is they really like the ability to bring in software tools from various places all into one place in the g.nome canvas, and have them presented as tiles and visual elements that they can then change parameters and draw connections between tools, and really have access to all of that open source environment to build and modify workflows. So they have access to Seurat right in the canvas, and they don't have to load up a coding environment. They can do it in Canvas's in g.nome's no code environment and just build those workflows, or actually modify the pre-built workflows that Nora has built for our customers.
Mm-hmm. Yeah, and I know it's something that I hear quite a bit from our customers as well, right? As they just struggle with getting the analysis done. And so it's great to hear that we have solutions in place to help with that.
Yeah,
so as a follow-up to that, Nora, are there any breakthroughs within the single cell research that you are most excited about?
Yeah, so as an engineer, I think what's always exciting to me is seeing how technology develops. Single cell really only came about in 2009, and since that time, we've seen just so many advancements. And so it's always exciting to me to see just the field accelerating at a really fast pace. And I'm always excited to see how people take a technology and then adapt it and get really creative with it. So one that I'll mention in particular that I thought was exciting is the ability for live sequencing. So one of the limitations of single cell is that we capture it at a static point. So we only see gene expression from one point in time, and the performance of the analysis or the processing of those cells kills them. But live sequencing allows us to study cells, in real time, basically by repeated sampling of the cytoplasm. And that opens up, a couple doors for us. So we can start looking at changes in expression over time, which help us understand response to stimuli, which is relevant in drug discovery, as well as understanding, the trajectory of a cell over the time course of its development. And then another one that's a bit similar is lineage tracing with single cell, which is where a cell is engineered with an inheritable barcode. So all of its progeny inherit that same barcode. And that allows us to, trace the lineage of a cell, and it's relevant to developmental biology. So we can learn how complex organisms develop from individual cells through this barcoding, which I think is very exciting.
Definitely. Thank you for your perspective on that. I think another huge component of single cell, right, is the analysis. And we've both kind of touched on this in that there's a lot of moving pieces. There's large data sets. There's complexities when it comes to analyzing these types of data. Nora, can you explain, some of the pain points that researchers might face when they're trying to analyze these complex data sets?
Yeah, so I'll just talk about a few things that I feel like I experience all the time when I'm working with single cell. Probably the first and most obvious is just the sheer data size and the complexity of the data. So single cell data sets are typically very large. If you think about it, you're basically taking, thousands to hundreds of thousands of replicates that are all getting sequenced. So that's just a massive data set. And then also, as cost per cell, decreases, data size is increasing even more. And so that makes storage and processing pretty computationally intensive. So you have to have architecture in place for that. And then another big one is standardization. So as single cell has grown more popular and more pervasive across different fields, we've seen more platforms for creating the data, and then technologies and softwares for processing the data arise. And I think kind of an interesting little number is that since single cell was developed in 2009, we've seen the number of publications using single cell grow exponentially. So in 2020, there were 2,000 publications which generated single cell data using a number of different technologies. And when the technologies, or when the kits are different and when the software for processing is different, it really can start to complicate how we compare studies and how we integrate data, and it can affect, reproducibility of analysis. So I think standardization is a big problem, and we need better kind of architecture for standardization. And then cell type annotation is a big one that I'm sure a lot of biologists experience as a pain point. At the end of a single cell analysis, the goal is to cluster your cells and identify what those cells are. So it really doesn't mean anything if we aren't actually able to identify and annotate what those cells are. And there's a few methods to do this, which can involve varying levels of manual and automated annotation. So with manual annotation, it really requires a user to know the marker genes that are associated with their cell types. And automated annotation can take a couple different forms, but regardless of which form it takes, it does require some sort of reference or training data set, meaning that there has to be previously established data sets of cells and their associated annotations by which you can compare your own unannotated data. And as you can imagine, this would be very difficult if you don't know what you're looking for in your experiment, or if you are trying to identify novel cell types for which there's no good reference. And I think this kind of echoes our need for a comprehensive cell atlas and for strong data sharing and standardization practices, so that people just have access to a broader database of broader resources by which to do their analysis.
Mm-hmm. Yeah, and I think there's definitely a lot to unpack here. You mentioned things around data and complexity, as well as trying to annotate some of these cell types. Kit, it would be great to get your perspective on this and how g.nome might be able to help address some of those things that Nora talked through.
Yeah, absolutely. And I think, as a scientist, the first thing that you do after you run one of these big NGS experiments is you're like, what am I going to do with all of this data? And it's this whole new problem that you have to solve. Fortunately, and even with the basic IT infrastructure, it's still difficult to figure out where to put it and how to analyze it. Fortunately, Genome, offers a single place to store, process, and analyze, all different types of NGS data. And so, you're able to kind of easily move the data to there and process it. In addition, with all the publications, as Nora said, there's so many different tools and standards that have been developed over the last 10, 15 years for genomic data. It's hard to know which ones to run, and converting between different standards and file types can be very daunting. And it's nice to have a single place like Genome where users can do all of that in a single environment. And so we're really reducing the complexity of single cell analysis, but providing a lot of these tools in the environment and these pre-built workflows with popular tools such as Seurat, so that you can cluster and annotate your cell populations with data from other places. And so it makes it a lot easier to move to the point of interpretation. So you're just getting to the point where you're modifying workflows or swapping in different tools, and you're able to quickly go and make those comparisons and analyses for your experiments, and actually make comparisons like control versus treatment groups or time series, and kind of gain that understanding from your data as quickly as possible.
Nora, from your standpoint, is there a feature in Genome that you are most excited about?
Yeah, so I think what's always exciting for me is when I and we as a team are able to take something that I previously experienced as a real pain point in doing analysis, off of platforms, so off of Genome, and then we bring it onto Genome and we're able to resolve that issue for the user. So one example of this is processing really large data sets. I recently analyzed a data set with 26 samples, 130,000 cells, and just wasn't able to do it locally. As your compute resources become more limited, it gets really challenging to integrate 26 different samples. And so I was excited about how we handled that on Genome, just to make, we don't want the user to worry about compute resources allocation and things like that. It's just, I want to be able to run this big data set without it, failing when it hits a certain point. So I'm really excited about that and how we've built in parallelization as well. So if you're processing lots of data, it should take the same time as just processing one sample, because all of these steps are parallelized. So I think that's one that I'm really excited about. And then as someone who loves data visualization, I'm really excited about the reporting that we brought in. So it's so important to be able to create figures that are easily digestible and understandable, and to be able to share your data with coworkers or your manager. And so I'm really glad that we've brought that capability onto Genome as well, that every time you do an analysis run, you get information about the QC and the clustering and feature visualization and things like that our users are really interested in, just all in one concentrated place.
Yeah, and I know I'm really excited about what we've done with allowing researchers to actually customize the workflows. That was something that I would hear a lot from our customers, is that either it's too simplified, right, and that there's no ability to actually modify the workflow, or it's too complex and that you have to know command line and code. And so something on Genome, of course, right, is that you can do things like batch correction or normalization, or filter out some of those cells, which is, of course, super, super important to the researcher. So I'm very excited about just how the workflow has been built itself. So thank you both for your perspective on that. And we talked a lot about some of the challenges. I think one of the other things that we hear a lot in the space is artificial intelligence and how this is revolutionizing drug discovery. It's a word that's thrown around quite a bit. So I'd love to get your thoughts, Nora, on the role of machine learning and AI as it relates to addressing some of these challenges around single cell, maybe as it relates to clustering or automated cell type identification.
Yeah, so single cell is really the perfect application for machine learning, in my opinion, because we start with a very large high dimensional data set, and the goal is to detect very subtle patterns and differences. ML is already critical to single cell analysis, whether we recognize it as that or not. But we use unsupervised ML algorithms like t-SNE and UMAP to cluster cells based on gene expression patterns already. So what I'm really excited about is how I think we're going to see foundational models start advancing single cell analysis. And when I talk about a foundational model, what I'm referring to is a model that is trained on a broad base of data, and it's capable of diverse tasks. So these models can serve as a foundation upon which to build more specialized models. An example of this is SC-GPT, which came out of the Bo Wang lab at the University of Toronto. And their foundational model is valuable because it can be tweaked to fit a variety of downstream tasks, which are really pain points in single cell analysis. So it can be tweaked for cell type annotation, batch integration, as well as multi-omic integration. And their most updated model was trained on 33 million cells from various tissue types. And when it was tested on a cell type annotation task, it was about 85% accurate compared to manual annotation, which I think is already pretty good. But as the field advances and we get more and more and more public data out there, our training data set basically grows. And so we can see that the accuracy and the capability of these models is only going to increase. So I think that's something that's going to be really exciting to watch, how that affects the field over the next 5 to 10 years.
Mm-hmm. Yeah, and Kit, I'm sure that this is something that your team has thought about. What is your take on this? What do you think is the greatest potential for AI and ML as it relates to which I'm sure is kind of a $1 billion question.
Yeah, absolutely. I think our ability to generate data has far outstripped our ability to analyze and interpret the data. We, especially as sequencing becomes less and less expensive, we're just doing more and more genomic experiments. And so the real exciting thing about these foundational models is that we can teach them basically the language of nature, and we can use them to pull out insights that would otherwise take us years to figure out. Single cell GPT is a really good example of this. It'll take the laborious task of drawing gates around cells and make it easy. And this is one of the reasons why I think g.nome is so powerful, that it lets our users take advantage of these cutting edge tools, right? And they can bring them into the platform, and they can either build a whole new workflow, or they can tack them onto a workflow that they already have, or replace an existing tool. And they have all that flexibility, as well as the power of these brand new AI ML tools. So it's really exciting.
Yeah, no, it definitely is. And I know I hear it quite a bit when I'm talking to customers. They're always asking about AI and ML and how it relates to our software. So I think for kind of the remaining time of this discussion, I'd like to focus on what's next and where we're heading in this space. So Nora, how do you think single cell is going to expand and what needs to be done moving forward to make sure that we're meeting the needs of our customers?
So I think the most obvious is probably just higher throughput and scalability. So as cost decreases, we're going to see people doing larger and larger experiments with more cells. And so technologies, both biological and computational, need to be even more high throughput to handle these data sets. And that's going to require automation, scalable data storage, and scalable processing methods. And then also multi-omic integration is a big one right now. So we're moving towards integrating multiple types of data, so genomics, transcriptomics, proteomics, all at the individual cell level. And the complexity of that is a major obstacle. It's going to require development of new technologies and pipelines that can capture and analyze multiple data types simultaneously. And I think also integrating that volume of data is going to require significant computational resources. And I think that also kind of leads to my next thought, is just on standardization and reproducibility. So as it's becoming more widespread, what I would really like to see is better standardization to enable data sharing and reproducibility, because there have been so many times where I take a public data set and I'm trying to go to their methods and figure out what they did, and it's always like next to impossible because there's just not quite enough information there. So I think that's one of the things that I really value about g.nome, not just as an engineer, but as a user of it all the time, is that I can go back and see every run, what are the exact parameters that I used. And I think tools like that are going to help the field move towards better data sharing and reproducibility. And then I think the last one that's becoming big is spatial transcriptomics. And just as we saw single cell start to become more standard and more people using it across different disciplines, I think we'll see the same for spatial transcriptomics because it allows you to capture not just the gene expression data of individual cells, but to see where those individual cells are located in space within your tissue, your sample, so that you can start to explore questions about the microenvironment that cell is in and how it's communicating with cells around it, which will be really exciting to see, the discoveries that are made from that.
Yeah, and I would agree definitely with all of those points that you touched on from a customer standpoint. That's what I hear all the time, especially around multi-omics. Researchers are wanting to process not only their single cell data, but they want to start to combine it with their attack-seq data and look at their gene expression data with other types of data or public data, right? And so I think that that's going to be a really important component of the analysis. And then, of course, being able to reproduce that, right? That's what researchers always want to do, is do that moving forward. And then lastly, you touched on spatial, which I know I'm super, super excited about. And I know Kit is excited about as well. It's kind of the newest thing on the horizon in terms of adding context to the single cell data that you're analyzing. So Kit, would love to get kind of your perspective on this. What does that mean for us and for our users that are going to be on g.nome?
Yeah, absolutely. So, spatial biology is such an important addition to single cell genomics. And, instead of just, inferring cell-cell interaction networks through gene expression patterns, you can actually look at those cellular neighborhoods and kind of piece things together about, how one group of cells at a certain location are affecting another. I was at City last weekend, and it was some great talks about how the environment around a tumor acts as a physical barrier preventing the immune system from destroying the tumor. And we can only start targeting those cells and changing that environment if we know where they're located and how they're interacting together. And it has so much promise for medicine and research. But notably, spatial biology is a magnitude greater in data storage and processing needs because it's just so much more information, spatial data. And so, that's where cloud computing is so important. It's already important for single cell, and it becomes even more important when you layer on spatial data and then try to integrate all of those different data types together. And so, the great thing about Almaden Genomics, we're providing all of that cloud infrastructure to our users to process these large data sets so that scientists can really just focus on the data analysis and not spend time managing the cloud accounts or a computing cluster, and they can just focus on trying to gain as much insight from their data as possible.
Yeah, and I think as a follow-up to that, I know at Almaden Genomics, we're really focused on becoming that end-to-end solution for our customers, right? Like right now, we can help with processing the data, whether that's in-house data or publicly available data. We're also working, of course, with the partners on the upfront portion of the analysis, so those curated data sets that can be brought in to supplement, as well as the downstream. So that's the predictive modeling using analytics and really trying to be that end-to-end solution. Can you talk a little bit more about the future of g.nome and Almaden Genomics and what this is really going to mean for our customers?
Yeah, absolutely. We have unique solutions today that allow data scientists, benchtop scientists, and scientists of all types to collaborate and analyze large data sets today. We have our pre-built workflows that are available to our users where they can come in and analyze their data on very reliable and robust informatics pipelines. And we're also building out brand new functionality. As Nora mentioned, we have a lot of figures and graphs and tables that you get as part of your analysis. But we're also building workflow reports, right? Where you get all your data in one report. And it generates those useful figures. So you have a detailed layout of all the critical results for a single experiment. I'm really excited to get that in customers' hands. Finally, we're always working hard to bring in new open source tools into the environment, especially in the realm of AI and ML, and integrate these new data types such as protein and epigenetics and spatial into genome workflows. So I think the future looks really great for the users on who are analyzing data on the g.nome platform.
Mm-hmm. Awesome. So thank you both for your perspective on everything as it relates to single cell. It looks like we do have some questions in the chat box. For those that are still hanging on, if you have questions, feel free to type them in the chat box. But the first one we have is, do you have workflows in place to process publicly available single cell data?
Yeah, so we're right now bringing in a workflow for integration of data from SRA. So that's raw FASTQ sequencing data. And so that will allow users to take the raw FASTQ data, pull it in just with the click of a button on g.nome, and then run single cell analysis from there. And then we're also working on the same for GeoFetch. So Geo is where people typically host, count matrix data. So they've already aligned their data and they've gotten to the point of a count matrix. And so we're going to build the same pipeline for Geo. So what's currently available is SRA, and then what will be available is fetching data from Geo, because often you just want to start from that point, or you potentially don't have access to the raw FASTQs. So we want that to be accessible as well.
Awesome. And then another follow-up question is, what visualizations are users able to create on g.nome?
Yeah, so starting with QC, we show users the distributions of quality metrics, such as the number of features of number of unique genes, as well as number of UMIs in their count data, and then also the distribution of mitochondrial DNA. And then we also show them kind of scatter plots, or combined scatter plots of those same metrics as well, which can help them just identify patterns in the data. And then we also show them the top highly variable genes expressed within each sample. And then in terms of clustering, we show them either a UMAP or t-SNE visualization, whichever has been chosen. And then we also show them the top expressed genes per cluster. And then we show them the feature visualization. So they can go in and examine a specific very useful for cell type annotation.
Awesome, thank you. And it looks like we have one last question. How important is data quality and accuracy in single cell analysis, and what steps can be taken to ensure the reliability of the single cell data?
Yeah, so QC is absolutely essential. I think, speaking from experience of trying to reproduce analysis from public data, if you don't know the filtering parameters that you used, you are not going to get lucky with your analysis. You're probably not going to happen upon that if there's hundreds of different options that they could have used. So it's really essential to know what QC parameters were used to filter the data, and I think because that will just completely change the results of your clustering. And I think it's steps that need to be taken to ensure the reliability of that. It's kind of what I mentioned before, is that you need really good logging of everything that you're running. And as a bioinformatician, if you're doing all of that work yourself off platform, you really need to be accountable and keeping good track of what you're doing. But if, when I run things on platform, on g.nome, that is all handled for me. So it's really easy for me to just go to my runs and see exactly what ran, what parameters I used to get kind of the outcome that I desired, because we know that really one of the needs in single cell analysis is for iteration. So you might spend 80% of your time iterating and trying to figure out the right parameters for your data to arrive at good clustering, because every data set is so different, and it's unlikely that just the default settings are always going to work for every data set. So it's really important to keep track of that type of information. And yeah, it's nice to have that handled for you.
I think that is it with questions, unless there's any last minute questions coming through. Thank you, Nora, for your input on this. It's great to hear your perspective on single cell. Kit, you of course as well. It's great to hear your perspective as it relates to g.nome. And thank you everyone so much for joining. If you have any follow-up questions, feel free to email us. We will send out this recording for everyone that registered and attended. So thank you so much, everyone, and have a good rest of your day. Thank you.
Thank
Bye.
Thanks, everyone.